for keeping us on time. All right, our last speaker in this group of case studies is Carolyn Nagler. She is the Bunning Food Allergy Professor and Professor of Pathology, Medicine, and Pediatrics, so she basically runs the whole place at the University of Chicago. She's an immunologist, and her main focus has been on mechanisms of tolerance to dietary antigens, and she'll talk about the microbiome and food allergy.
Thank you. Can you hear me clearly with this microphone? I'm really delighted to be here today. So I'm not going to talk about infectious disease. I'm going to begin by introducing the problem, and that's the dramatic increase in prevalence of food allergies. So I would guess that everyone in this room knows somebody that has a food allergy. And in fact, this is data from 2019. It's now up to 32 million Americans with food allergies, 1 in 10 adults, 1 in 13 children, a 377% increase between 2007 and 2016. When I was in school, my brothers and I ate peanut butter and jelly for lunch on most days. My children, as they moved through elementary school, were, by the time they finished elementary school, were in peanut-free classrooms. So this is a generational increase. And how do you explain such a dramatic generational increase? So we turned to the microbiome, and I'll define the microbiome very simply as the microbes that populate our skin and mucosal surfaces and have beneficial functions for our health. I will refer to microbiome and bacteria interchangeably in my talk, but remember that the microbiome is far more complex. It includes all kingdoms of life. It's just that we understand the bacteria most clearly at this point. And the hypothesis has been that something about our 21st century lifestyle has changed the composition of our commensal bacteria in a way that's detrimental to our health and is promoting these non-communicable diseases that we've discussed. So this is from a recent review that I wrote, and it emphasizes the influence of the microbiome and food allergy. But in fact, I could list on this side of the slide obesity, diabetes, autism, inflammatory bowel disease, many other non-communicable diseases that have been linked to changes in the microbiome. So what are these lifestyle culprits? Antibiotic use is far and away the biggest offender, not only prescribed antibiotics that are given very commonly in the United States, for example, for viral ear infections early in life, but also exposure to antibiotics in our food and water supply. Diet is another big influence. We've co-evolved with our microbiota over millennia. Our ancestors were not eating McDonald's. They were eating a diet high in fiber, low in processed foods, low in sugar. Our bacteria eat what we eat. We've changed their food source. We've moved from situations where we were in close contact with bacteria from our environment in rural and suburban settings to living in sealed houses in urban settings. Vaccine-induced immunity and reduced exposure to infection, particularly for this group, it makes the interesting point that these diseases have increased as the prevalence of infectious disease has decreased. And eradication of previously common enteropathogens like Helicobacter pylori and gastrointestinal helminths. So all of these factors together, in the setting of genetic susceptibility, we're proposing are driving the prevalence of food allergy. So to begin to address this, we teamed up with a group at the University of Naples, and our collaborator there gave us just 20 fecal samples from healthy infants or cow's milk allergic infants at 4 to 5 months of age. And it was very fortunate that we set up this collaboration because this is a much more homogeneous patient population than you'd see in an American clinic. Everybody was Neapolitan, there was no racial diversity, no ethnic diversity, they were eating a similar diet, and that's why I think we saw a very clear difference. So at 4 to 5 months of age, the healthy infants had the type of bacteria we expect. They were dominated by yogurt type bacteria, our conventional probiotics, lactobacilli bifidobacteria. These were depleted in the cow's milk allergic in infants, who instead had an adult type microbiota, as if the normal ecological succession that populates our microbiota from birth through the first few years of life had gone forward at warp speed. So we wanted a way to really study the interactions of the bacteria with the host. And in food allergy, unlike some other diseases, we have no access to biopsy samples. So we thought it was very important to make a reliable animal model that we could use to study these interactions. So the approach that we took was to use germ-free mice. We're lucky at the University of Chicago to have a state-of-the-art germ-free mouse facility. So the way this works, if you haven't seen it, this is a flexible film isolator. You can see here a couple of individual mouse cages. Everything that goes into these isolators has to be sterile inside the tube and outside the tube. Everything has to be controlled. The mice live in these isolators for their entire life, and every manipulation is made by reaching in through these heavy gloves. So it's a cumbersome methodology, but very, very powerful, because you can control, if you can identify the populations of bacteria you're interested in, you can precisely control them and introduce them into groups of mice. So the way that we decided to approach this is that we got fecal samples from 4 different healthy donors and 4 different cow's milk allergic donors, all 6 months of age, all controlled for all the demographic variables we could identify. Each one of these donors gets its own isolator to keep the microbiomes of that particular individual intact. We found the first time we did the experiment, we transferred fecal samples from breastfed infants, and we found that we didn't get a perfect translation of the fecal material from the humans into the mice. And we discovered that what we were missing was bifidobacteria, which is a breast milk dominated bacteria. And that made us think, well, what we really need to give these mice to get a good transfer of the human bacteria into the mice was formula. So we fed the mice the same formula that the infants were consuming. And this figure represents results from 100 gnotobiotic mice. And I think it makes our point pretty clear. The mice that were colonized with the healthy infant microbiota are protected from an allergic response to food. We measure allergy in mice by a drop in core body temperature. We measure anaphylaxis. The mice that have germ-free, that have no bacteria at all, are susceptible to anaphylaxis. The mice that got the cow's milk allergic CMA microbiota are susceptible to anaphylaxis. And this symptomatic readout of anaphylaxis correlates with the induction of an antigen-specific, so this is bovine lactoglobulin, IgE, IgG1, and mucosal mast cell protease 1 response. We spent a lot of time identifying the bacterial populations and then also looking at how these differentially abundant operational taxonomic units, so bacterial sequences in the healthy or CMA colonized mice, how they could be divided into particular groups and how those groups related to gene expression in the ileum of the colonized mice. And to make a long story short, what we found that there was, when we looked at the integration of those 2 data sets, that there were 9 OTUs that significantly correlated with genes upregulated in the ileum of healthy or CMA colonized mice. And of these, we saw that 3 of them were from the family Lactospiraceae, which we had identified previously as allergy protective. We looked back to the database and found that the closest match for those sequences was a bug called Anorostipes caseae, which was a known butyrate producer in the human infant gut. So we then transferred A. caseae by itself. We monocolonized the mice. And we did this actually at the request of the reviewers. And we found, to our surprise, that AKCA by itself protects against an allergic response to food using the same parameters that we measured previously. I also want to point out for this group that in other work with Gabriel Nunez, a collaborative project with Gabriel at the University of Michigan, we also showed that bacteria from this class, the Clostridial class, which contains the Lactospiraceae, are important for protection against colonization with bacterial pathogens early in life. So the picture that emerges then is that this is a population of what's been called the gut's peacekeepers. Bacteria like anerostripes, or in the colon, Faecobacterium prausnitzii, ferment dietary fiber to make short-chain fatty acids, including butyrate, which have important effects on maintaining the healthy epithelial barrier function with an intact mucus layer that keeps opportunistic microbes and toxins and food from gaining access to the rest of the body and causing disease. So we are interested in developing microbiome modulating therapeutics to prevent and treat allergic or inflammatory disease. To that end, I've co-founded an academic startup company with Jeffrey Hubble, a professor of engineering at the University of Chicago. And our first approach is to make synthetic polymers to which we have conjugated microbial metabolites. The first candidate is butyrate, but we're also considering live biotherapeutic approaches and prebiotic dietary fibers so that we present the bacteria with their substrate and all of the components that they need to be able to produce the metabolites optimally. So finally, I'd like to thank all of the people that did this work and our funding sources and answer your questions in the discussion period.